thank you very much indeed for the invitation to come to Ireland, and now you know why I always accept when I'm invited across. Sadly, that 1/8 Irish isn't enough to get me an Irish passport, which right at the moment, anyone I know who's got Irish connections has been doing that. But anyway, I'd enjoyed it when I found it out. So what I'm talking about today is trials, but I'm not talking about doing trials. I'm really talking about the use we make of trials and indeed other kinds of evidence to make decisions for, by, with patients. And what I will do as I'm talking is try to highlight what I think is some of the where we're using methodology and where I think there are some very open questions. So most people here will have some informal idea of what's meant by benefits and risks, but you may not really know how it feeds through. In particular, I'm going to be talking in the context of drug regulation, although I think what I've got to say has much more general applicability. So what I'm going to do to start with is to talk through a relatively simple example, just to get some language and concepts, and then I will tell you about a big project I was involved in for some years, which is answering or trying to set a foundation for a much more general way of approaching it that can deal with more complicated questions. But let's start at the very beginning. So I'm sure all of you at some time have either been ill, whether it's a cold or something more serious, or if not, you've had a parent or a child who's had a problem. And you have to decide what to do about it. You have to decide what treatment to take. And so if you think about a framework for formalising that decision-making, some work I did a long time ago actually with Adrian Smith, really they had all of these hieroglyphics and statistics, and I said to Adrian, how can we put that into kind of words? And what it boiled down to wasn't rocket science. You've got a decision maker, they can take some possible actions, there might be some uncertain consequences, you've got hopefully some evidence, which is what most of the rest of you are talking about how to collect well, but you can't make a decision without then thinking how you care about certain things. Formally, I've called that utility assessments, and we'll be spending quite a bit of time thinking about what we mean by that. So, the concrete example, because I always work far better in concrete examples. Drug, which is known by the generic name of trastuzumab, certainly in the UK it is known as Herceptin, and it's one of a class of drugs called monoclonal antibodies, which have really revolutionised treatments of a lot of diseases. And the background to this was that at the stage I'm talking about, it had already been given a licence for more advanced breast cancer, and the question then was it going to work in rather earlier stage breast cancer? And the reason it's a simple example is, for a start, breast cancer disease that many people at least know something about, and it has some relatively simple outcomes. But secondly, there was just one trial, so again, it's good for illustration. It is, of course, for a particular group of women. I think in breast cancer they were doing personalised medicine long before anybody actually invented the phrase, so we'll take it as read that this is a woman. We're talking about a woman who's got the right kind of HER2, who'd done the right things. So what did the trial evidence say? Well, the basic thing that you want is to be disease-free. I mean, you absolutely don't want to die, but very small numbers actually die. So on placebo, it's about 2.5% at 2 years and less than 2% on active treatment. But, so really the aim of this treatment is to have people disease-free at that, or at least no more disease progression. And that, under this treatment, goes down from 22% to just under 14%. So that's good by anybody's standards. But on the other hand, there is cardiotoxicity, problems of the heart, and a lot of drugs for cancer actually have this characteristic, but it is something you have to weigh up. And for the serious ones, that goes up from about 6 in 10,000 to about 6 in 1,000, whereas for the less serious ones, it goes up from about 1 in 200 to 3 in 100. So you could say, gosh, tenfold increase in risk. That's how many of our newspapers would do it, and that sounds awful, but actually the numbers are quite small. So the question, if you're the woman deciding whether to take Herceptin or not to, so a very stylised decision, you're really weighing up these risks. And I don't think we need much in the way of formal statistics. I think many people with those numbers, they might need to ask to get their heads around what the numbers are. But I think confronted with those numbers, many women would then say, yes, I know what I want to do. But nevertheless, I'm going to formalise it because it sets us up with a framework and a language. So, in terms of the framework I've just shown you, the decision maker is the woman. She may choose to talk to the partner in her life or her children, but basically it's her decision. She might even talk to a doctor. Yeah, quite a lot do. Her decisions, for the purpose of keeping it simple, are to take this active treatment or not to take it. Of course, in real life, she might be saying, what are my other options? What are my other drugs I could take? Can surgery alone do it? But let's just keep it really simple at the moment. And the uncertain consequences, the things that she's trying to weigh up that are relevant to this decision, are the breast cancer recurrence, death, and this cardiotoxicity. We've got a nice, simple source of evidence, because I chose the trial, I chose this particular example to keep it nice and simple. And what I mean by utility assessment is how you balance that disease-free survival and cardiotoxicity. Now, I first came across this example when I was doing, for many years of my life, I did drug regulation, and it came through like that. And we didn't formalise it, but then later on, when it went through, it comes first the UK and then goes through for European regulation, and it started to get more formalised at that point. But the kind of things we might use, and again, it looks nasty when I write it as 1 over delta, but we could use our old friend, the numbers needed to treat. What's the number of women we need to treat to observe a benefit? And we have, Stephen Evans says there should be numbers to need to treat to harm, but I can't say that, we'll do it as numbers needed to harm. What's the number of patients to treat before you would see an extra adverse event? So those are 2 metrics that are around. They're very much liked certainly by some doctors. They're very much loathed by some statisticians. They have some dreadful statistical properties, but they are measures that we could use. So if we do those calculations, the numbers needed to treat is 12. So for every 12 or 13 patients treated, 1 will benefit from progression-free survival. Right, so it's certainly a benefit, but perhaps slightly less dramatic than you might have thought. The numbers needed to harm is just under 40. So for every 40 patients treated, 1 will get cardiotoxicity. So the question is, how do we weigh that up? And certainly, you sort of feel that numbers needed to treat less than numbers needed to harm, which is the case here, is probably a good thing. But that is making a sweeping assumption, which we will come to. So when the MHRA did their formal assessment report, what they said was if disease-free survival and primary cardiac events were combined into a single endpoint, so some kind of messy composite endpoint, it will be dominated by disease-free survival, and the hazard ratio favours the active treatment. So that's basically another way of saying numbers needed to treat is less than numbers needed to harm. But the big, huge thumping great assumption that I'm sure many of you have spotted is that that is saying that breast cancer and cardiotoxicity are equally important outcomes at 2 years. And many women wouldn't think that. Now, I think for many women, actually the breast cancer probably trumps the cardiotoxicity. Even if they were equal, they would actually say, yes, I would take the drug, but that's a personal choice. And so one of the questions is, does further quantification add anything to this scenario? And one way of looking at it would be to say, what kind of weighting would you have to have in favour of or against the cardiotoxicity before it became such that you would not take this drug? Because there's many drugs that we do not take because of the risk of very serious side effects, and they get pulled from the market. And indeed, we will be discussing one in a little while. So what that example, I hope, gives you is some sense that you're making a decision, that you've got some evidence, you've got some uncertainties. I mean, I needn't talk to this audience to say that we could put confidence intervals and all the rest of it around those outcomes. But the key thing is that you have to weigh up, and you might or might not want to assume that everything is equally serious. And if you don't, it begs questions of how you could do it formally. Now, before we leave that example, I want to make just a point about decision makers, and we might return to this in discussion. I draw it as a circle, and I put patients at the centre, because if it's not about patients, I don't know who it's about. But patients are actually constrained by what their healthcare providers will let them do. And or what they make available. The healthcare providers are constrained by what health technology assessment types, people like NICE in the UK, I'm not sure what the Irish equivalent is, but I'm sure you've got one, basically bring cost effectiveness into the argument. They are constrained by what regulators do. And they are constrained by what the companies do. So they can't license something unless a company's trying to make it. And just to show you how that works with the example that I've just given you, some of you may be old enough to remember when Herceptin was over all, you're a journalist, aren't you? Was over all the newspapers. And it was shock horror, women dying because they are being denied an important new treatment. That was the tone of it. Well, first of all, I've shown you the data, and I think it's not true that women were dying because by and large, this was early stage breast cancer, and actually they weren't dying of it. The reason they didn't have access to it was their healthcare providers didn't yet have it on their lists of available treatment. They couldn't put it on because in the UK, at any rate, NICE hadn't pronounced on whether it met the cost-effectiveness thresholds. NICE weren't being slow. NICE are not allowed to look at drugs, at least they certainly weren't then, until they had a licence. The reason the regulators hadn't given it a licence wasn't because they were being slow either. I mean, they're not always the most efficient people, although they are now because they've got timelines. The reason they hadn't looked at it was the company concerned hadn't yet made their submission. And regulation, at least for new drugs, is a reactive process. It's a bit more proactive when there's a safety concern. And basically, the company were finalising their submission, to give them the benefit of the doubt, to get it right first time, whilst all of this publicity was going on. As soon as the company put in the submission, the MHRA were ready for it and expedited it. NICE then looked at it and said it's cost effective. Hospitals could put it on their list, patients could have it. But what that illustrates, and we will drill down into, is that the decision-making involved in medicine is different for different groups, and the considerations are different as well. So the kind of things that drive a decision about whether it's cost effective aren't exactly the same things that will drive what a patient does. But broadly speaking, the evidence base is the same. So, a little bit more about drug regulation. The national regulatory authorities work through the European Medicines Agency, of which the UK is a part still at the moment, but sadly will not be after next March, which is losing a huge amount of money because it works by devolving business, and the agency is moving. But essentially, the EMA acts as a sort of people now apply jointly to Europe. And the FDA is probably the best known, which is the US one, but most other jurisdictions have licensing. They're either part of one of those or they have their own. And when I first got involved in it back in the very early 90s, it was effectively under our Official Secrets Act. I don't think I had to sign it, but you were barely allowed to say you were on the committee. You certainly couldn't talk about the business. And the world has changed in many ways for the worse, but in some ways for the better. And it has become much more transparent. And so we have to defend what we do. And I have to say that all the time I've been involved in drug licensing, I think those people around the table have really tried hard to make the best decisions. But now They are put out there. That's why I could show you what was in the MHRA public assessment report, for example, and you can critique them and say, I disagree, or you can say, okay, now I understand that's how they're thinking. Maybe we need to do our trials a bit differently or choose a different endpoint. So we need to be able to explain to other researchers, but also to patients and other stakeholders, which of course in the pharmaceutical industry is a major one. So the question that was bubbling up sort of 10 years or so ago, probably 15 years, was on both sides of the Atlantic, was can more formal approaches to decision-making help us do this better, help us make better decisions, help us explain those decisions? And there was a sort of subsidiary question, which I won't really go into today, about whether more modern methods of graphical display might help, although some of my slides, you'll see some of the fruits of that. And we were by no means the only people doing it. The European Medicines Agency had already had a bit of a project on this. There were some lovely acronyms around. There's BRAT, the Benefit Risk Assessment Task Force, the other side of the Atlantic that turned into UMBRA. Various groups like ISPA, which is one of the big societies, had a group on it. And the one I got involved in is the one at the bottom of the list, which is something called the Innovative Medicines Initiative, which is a big sort of public-private partnership between the European Commission in research funding mode and then with equal funding from pharmaceutical industry. And I got asked to lead a project on benefit risk integration and representation as part of that. Just to give you some sense of the scale of the collaboration, and this was just the project I was involved in, other pharma were involved in other projects, you can see that side, sort of Merck, AstraZeneca, GSK, Novartis, Pfizer, Roche, and I haven't read them all out, but basically most of the major pharma are involved in this. And then the public partners were, I won't read out the initials, but that includes the UK Medicines Agency, the Spanish Medicines Agency, the Danish Medicines Agency played a particularly large role in this one, a few academic organisations, and probably most importantly, IAPO, which is the International Alliance of Patient Organisations, representing the patient input. So I had to lead a collaboration of this little lot. I have to say, we probably spent the first 18 months just getting our language straight and realising that we weren't using it in the same way, working out what the scope of the project was. It was a commissioned project, so it should have been clear. But it was a 5-year project, and that 18 months we spent sort of talking and negotiating paid dividends later on. So what did we do? And I'm just giving you some highlights here. We did a methodology review. I've been talking when I was looking at the posters to one or two people doing methodology reviews, saying how hard it was. And we did a review of what quite a lot of other people had already done reviews of methodologies for benefit risk. So we didn't start from scratch. We read these reviews. And that's what it felt like. I mean, I'm pretty, long-serving methodologist. I went, whoa. And what didn't help was some of them had them in alphabetical order, which made no sense at all. So my very great colleague, Sharul Matiza, led this. And essentially what we did was to take that lot, and I said, I can't cope with that. I've got to have some structure. I'm a statistician. So we did magic. When I say we, that's the royal we. My team did the magic, and I get to come and talk about it. So the younger ones here will be beginning to realise that's how it often works. I have to say, I've made sure they've gone and talk about it as well. And what we did was to basically divvy it up into several sets of kind of techniques. So the first bunch over here are frameworks. The one I showed you, which was the one I did with Adrian Smith, was a simple version of a decision-making framework. There are others, some of them not very dissimilar, but they've got wonderful acronyms. The second bunch is things that kind of measure the outcomes, and some of them help you to trade off. So the numbers needed to treat and numbers needed to harm are in there as potential contender methodologies. The third bunch of stuff I was mystified by. There were things in there, directed acyclical graphs and confidence profile methods. Now, some of you may know exactly what they are, and some of you will go, pardon? Basically, they're the statistician's toolkit, but they are really quite fundamental. There's nothing particularly benefit-risky about them. But on the other hand, they are very useful sometimes. So we put them in a box on their own and we put mixed treatment comparisons and indirect treatment comparisons in there as well because they're jolly useful and not just about benefit-risk. What was the smallest box and grew a bit during the project turned out, I think, to be the most interesting part. And in case you need a bit of a dose later or you're thinking about your lunch, that's where I think the open methodological challenges are. I think here we had a whole basket of methodology, and it's really how can we bring it to bear on this problem, but it was there ready to be used, needed translating into this applied world. Whereas down that end, I think it's a very open space, and I'll be giving you just a glimpse of that. So what we did, I'm not going to go through all of those. We picked on a handful and said, right, these are what we think are the leading contenders. The others are just sort of either Me Too's or otherwise clearly not appropriate. And what we did was say, these are the ones we want to test out. And I have to say, at this point, there was a relatively small group of people really engaged, really into this stuff, and most of the rest was sort of passengers feeling, it's methodology, don't really know how to do it. And so what I then did, and it was one of the brightest things I did, was we then recast, we were in various working groups, said, right, we need to restructure our working groups into case studies. And that energized the whole thing. And what I'm going to show you today is the results of one of those case studies that really illustrates some of this. But there are plenty more if anyone wants to follow them up. And at the end, I'll mention what those other ones are. Now, I have to give my standard disclaimer. This is just for fun. Anything I might say that could possibly be taken as a criticism of a decision that a regulatory authority might have made shouldn't be taken that way. And the reason for that is that our job was to test out methodology. I was absolutely insistent we used publicly available data only, so that if anyone here wants to take this, play with it, redo it, they can do. But of course, regulators sometimes have access to other information, possibly they've seen it on the submission wasn't successful, and so they have more insights that sometimes might quite legitimately change their judgment. And the other reason is we chose really difficult case studies. There's no point doing the easy ones, and so they are ones that are quite finely balanced. So that's the disclaimer. Right, so we're talking about another monoclonal antibody. And it's called natalizumab, and this was, quite a lot of the monoclonal antibodies turn out to be used for a variety of diseases, but I'm talking about it here specifically for the disease of relapsing remitting multiple sclerosis. And if you know anybody who's had it, or indeed if anybody here has it, you know that it's a disease with an unpredictable but largely kind of downhill course over the long run, and up till a few years ago, there really wasn't much in the way of effective treatments. So when natalizumab came along, this was seriously good news. As you'll see from some of the data, it really has got some benefits. And then, of course, what happened, it was approved in the US in 2004. They got on with it faster than Europe. And by 2005, side effects that hadn't been in the trials, but as soon as you do it in bigger populations, you start to see the rarer side effects. And something called, I should only say it once because I can't, progressive multifocal leukoencephalopathy. Basically, these drugs get across the blood-brain barrier and do nasty things to your brain and can be fatal. It only happens very rarely, because if it happened more often, the drug would never have got this far. But it was enough for the FDA to suspend the license. And there's a number of drugs that have been really exciting, side effect, and that's the end of the story. Only this wasn't the end of the story, because by this stage, patients were getting a voice. And there are certain diseases, I think, because they're chronic and tend to affect younger people, where patients have got better organised. HIV is another one. And so the patients said, yeah, we know it's got a rather nasty side effect. It's also a rather rare side effect. Don't you dare take it off the market completely. Put it on the market, give us the information, and let us be the judge of that. I mean, other patients had said similar things about arthritis drugs and so on and got nowhere a few years earlier. But this, the FDA listened and they reintroduced it, clearly with lots of controls, looking at who is most at risk. And by the time they looked at it in Europe, that was available, it was licensed in Europe, and it's been reassessed many times. But it's still on the market, and we've learnt more and more about it. So what we decided for this case study was we would look at the benefit risk round about sort of 2009 when some of these were beginning to emerge. How do you weigh up the undoubted benefits against this really nasty side effect was what the problem we thought we were going to solve. So, just to show you some of the frameworks. So remember, the reason for doing this wasn't really to pronounce on natalizumab, it was to test out whether these frameworks were useful. So one of the lead contenders is something called PROACT-URL, Problem Objective Alternative Consequence Trade-off, and then other people had added on the uncertainty risk tolerance linked decisions. And this is something which has been used for many years. It's been used on things like where you bury your unwanted nuclear waste, which is another tough decision. So it comes from a different world, but has obvious application here. It emphasises uncertainty via sensitivity analysis rather than doing anything sort of more fully statistical. And another one that is roughly similar is BRAT, which was a sort of bespoke framework developed in the States by the Benefit Risk Assessment Team, which is a largely pharma-led. But essentially, they tell you the same things. They get you to step through, and the most important thing is to say, what's the decision context? Which harks back to that who's the decision maker question. I often hear a lot of muddled conversations about decision making, and nearly always you realise there's a muddle because one set of people are thinking about the regulators or the hospitals or NICE, and someone else is thinking about the patients, and they're not thinking clearly. And anyone who's done a trial knows if you don't have your question clear at the start, you get in a muddle with everything else. If you really think clearly what's your question, the rest you can work out, and it generally falls into place. So you say, what are you trying to do? What's the objective? What are the alternatives? So when I showed you the Herceptin, I just showed you against taking nothing or placebo, and I kind of use those interchangeably. But of course, and in our first wave case study, we just did natalizumab versus nothing, but then we developed this one on as a second wave case study, and we said, right, well, we've got competitors, beta interferon, glutamate acetate, are the other things that many patients would want to weigh up. And we didn't choose everything, but those were the ones that were relevant at the time. You want to know what the consequences are, which is essentially about your outcomes, and then some other things. So the important thing is that you spend time thinking this through. So those frameworks are used to sort of set the scene. Right. They, in themselves, are not particularly numerate or analytical, and actually, you can get a long way just by doing that, even if, for example, the Herceptin framework, you then don't actually formalise the analysis any further. One of the techniques that we were looking at, and it had some strong proponents, in fact, they were so strong that to start with, I was quite against it, because I thought this is just kind of special pleading, but actually my younger team said, actually, this is really great, it does what we need it to do. Multi-criteria decision analysis. And that is specifically there to deal with multiple conflicting criteria. Again, it comes from a background not just in health. And what it requires, and we'll go through with some examples, some probabilities, basically that come from data, which we like, we know about, we understand it. We haven't got it all perfectly sussed, but we're there. But the difficult things you need are things called utilities and things I haven't really talked about called weights. And again, I'll show you by example, and then I'll talk a bit more about where we get them from. And it's typically, when it's used, it's governed by PROACT-URL for structure and transparency, at least in the health world. But it is essentially deterministic analysis. It gives you an answer at the end, and all of that uncertainty that we're so used to talking about suddenly goes out of the window. So we'll come back to that. So to put that graphically, you need those three ingredients, and if you mix them together in a good principled way, what you'll get out is some estimates of the overall benefit-risk balance. So let's just go down this example a bit further. The benefits of natalizumab are that there's a reduction in relapse rate, because relapses are the thing that make you think, oh gosh, I can't get to work because my leg's gone, or your vision's gone, or whatever it is. And also a slowdown in the disability progression. None of these drugs are going to reverse it, but at least if you get a slowdown in the underlying progression, that's good. So those are the things that made patients say, this drug really is something that's useful. But there's actually a whole basket of side effects. The ones at the top are the more serious ones, the ones at the bottom are milder. PML is clearly the most serious, but it can produce seizures. If you're pregnant, and this is a demographic that might well be, it can produce congenital anomalies or spontaneous abortion. And then some other things, transaminase elevation may not mean much to you, but a liver doctor says, oh gosh, if their transaminases are going up, that may mean they're going to have liver problems further down the line. And flu-like reactions, which is not pleasant for a drug that you have to take in the long term. So actually, it's not just about the PML, which will come out. It's how we weigh these up. And then there's also the question of the convenience of taking it, because it's not a very pleasant reaction, but I've left that out of this in the interest of sort of keeping this reasonably straightforward. So we need some data. We really can't get any further than that without some data. And of course there are trials. These things would never have been licensed if there weren't trials. So that's good. And then the problems start. Because of course for drug licensing, what most companies do is my drug against placebo. Because to start with, that's the start of showing it works. I have to say that at the time the other drugs were done, there wasn't a lot else for MS. And so actually that was not an unfair trial to do. But what it means is, what I would like is interferon beta versus natalizumab, because if I was a patient, that's the choice I might well be making. We don't have the head-to-head trials. What we do have is trials, and notice they're over, not too bad, but I think life changes between 1996 and 2006. So those more or less reflect the licensing history. But in our basket of statistical techniques, we've got indirect treatment comparisons, if we needed them. This isn't actually, but you could have a mixed treatment comparison. And essentially, the simple version of this is if natalizumab is 4 times as good as placebo, and interferon beta is only twice as good as placebo, making heroic assumptions, you can sort of cancel and say, well, natalizumab must be twice as good as interferon beta. You're making all sorts of assumptions that the patients haven't changed, that placebo groups in the trials are comparable, and so on. But you can get an estimate, and it may not be ideal, but until someone gets around to doing the trial we really need, it's kind of the best we've got. So that's what we did. And the mixed treatment comparison can take in when you've got some head-to-head trials and the placebo-controlled trials. So what you get out of this is a colour-coded tables of data summary. And if you were doing this yourself, you'd be spending hours poring over these things, so I'm just trying to give you the general thrust. But essentially, the key thing about this is that everything needs turning into rates. There's nothing relative about it. So, for example, the relapses are nearly half the patients, 450 per 1,000, get relapses on placebo, whereas you can get that down to about a quarter on natalizumab. So you can say, well, that's a difference of 170 per 1,000. So the top line up there says that that's 170 per 1,000 because it's the absolute differences that drive the risk-benefit. You do the same for each of the other endpoints, and we started introducing, as part of the visual thing, some colour coding as to which ones go which way. And you can see, even if you can't see the numbers, and I hope you can't, because you've got yellow and blue, that's telling you you've got some things pulling you in one direction and some things pulling you in the other. So you've got a bit of a weighing up job to do. And those of you who've done this kind of analysis will know just how much sort of statistical jiggery-pokery there is to get to that stage. It may look simple as a presentation because it's numbers, but actually there's a lot of analysis underneath it. You then have to ask yourself how much you care about these things. And you have to do this for all of the endpoints. Again, I'm just showing you one to give you the sense of it. And this is the EDSS score, which is one of the standard scores for looking at disease progression. And basically, the best thing would be if nobody had it, and the worst thing is if 100% have it, so we arbitrarily call that 0 and 1. The difference between the groups is actually 11%. And so you can read off that the benefit of that is 0.89. Now, in itself, that's not terribly helpful, but it's a kind of building block. Did you ever play with Lego when you were young? Each brick in itself isn't much use, but when you put them together, you suddenly get something good. I've just bought my grandson his first Duplo. Love playing with that. Right, and then you combine it with a weight that we're going to talk more about, which is how much you care about that endpoint compared to the other endpoints. And then you get a benefit-risk contribution that, again, in itself is just, it's a slightly more complicated Lego building block, but you'll see how we're going to use it. So the first bit is, how do I care about this within? And the second bit is, how do I care between? And you don't have to assume a linear utility. You can draw funny shapes. I mean, if it's epilepsy and a driving license, it really matters that you don't have a recurrence within 6 months. If you do, it doesn't really matter where in 6 months. Sorry, I think it's 2 years for a driving licence, isn't it? You have to stay relapse-free. So there's a really sharp cut-off at 2 years, and your utility function might reflect that. But actually, I think in the scheme of things, that is not going to make a lot of difference. So we'll stick with the linear ones for now. And then you do some maths. You told me doesn't find statistics scary, and a lot of people find algebra scary. So I put that up to show that actually, for those that don't find algebra scary, it's just a weighted sum, but much better to turn it into something more visual. So what we've got here is, first of all, the weights, then the values, and then they are multiplied and added together to give benefit risks. And essentially, each line is one term, one component of that benefit risk, one for each outcome. It's rocket science in some ways. So the weights, PML is that great big blue one. That is by far the dominant outcome. If you were offered that you could avoid just one of these or choose to have the benefit, that's the one that you would chuck out first. We all would if we could. And then relative to that, relapse is a bit more important disability progression. We will come in a minute to where these weights actually come from. At the moment, just take these as an example of a set of weights. What the second one really reflects is the difference between the groups in the trial. And again, at the moment, this is just the natalizumab versus placebo for illustration, but you would be doing it for all your comparisons. The biggest one there is the relapse. That's why patients like it. It goes the other way for convenience. What about this? You can't see it because 2 in 1,000 minus 0 in 1,000 is invisible. And that's why most people just forget about it. If they're buying lottery tickets, suddenly that becomes really large because they think they're going to win when the odds are much worse than that. But actually, it doesn't register. And what that means is when you essentially multiply this column by that column, you get a big benefit risk component. You get that there, which is the downside of the infusion site reactions, and actually not an awful lot else is registering. And I think this is quite a nice mathematical or formal representation of what patients are doing. That's the decision they're making about whether they can tolerate this kind of, or the injections and what have you, versus the benefits. Actually, the PML is barely flickering. So that's an example of a benefit-risk analysis. Then what you do, and so I'm giving you a highlight of what was, of course, several months' work, and most of which was actually extracting the data from the reports and getting that together. That was by far the hardest bit. But once you've done that, and as coming out of your analysis, you've then got natalizumab versus placebo, natalizumab versus beta interferon, natalizumab versus glatiramet acetate. And those are stacked bar charts, which is where the colour coding comes in. And you can see that natalizumab dominates each of those comparisons. So it doesn't matter which drug you compare it to, whether it's placebo or the active. Under this analysis, with this weights, then it wins hands down. But this is still just a determinate, so that is essentially the stat graph, the one I showed you earlier. But it's still just a deterministic analysis. It doesn't mention uncertainty. So, there are, of course, a gamut of techniques for helping you look at uncertainty. My favourite one visually is the tornado plot, where you can explore sensitivity to weights. This one is looking at sensitivity to weights. You could look at sensitivity to the data or sensitivity to other things. And what that does is sort of it perturbs the weights you put in by 20% one way or the other and say, does it change the answer? And that scale, which is the overall benefit-risk balance in this particular example, standardised number of relapse, it's all positive. But what it tells you is that the assumptions on the weights that you're putting on the relapse is by far the dominant one. So if you really want to give some careful thought, that's the one you need to reflect most closely on. The next one is the infusion site reactions. And actually, the assumptions you make on PML and other things really aren't going to affect your decision. I mean, they're clearly, PML is important, but actually, the kind of way you've weighed that up is not going to impact on your overall benefit-risk balance. So, and in fact, I'm happy to make these slides available afterwards. There's a link there that if you don't like my weights, you can go in and you can tinker with them and do some, what if I think something different, which is one of the nice features of this work. And there are, of course, lots of other techniques for looking at sensitivity. But all the while I'm doing this, I'm thinking, I've been trained as a mathematical statistician. I spent 11 years when I was in Liverpool. I was partly in the medical school, but I was actually half in the stats department with, these proper Bayesian statisticians who know how to do decision-making under uncertainty, which didn't seem to be coming into it at all. And there's a literature there, well-known names to those in the world that you could go and look up. And essentially, the thing as a statistician I wanted to be doing was to extend the uncertainty analysis into a proper probabilistic model and look at the whole distribution. So I'll just give you a glimpse on that. And the idea there is that you would have not just a number for the benefit risk, but some sense of how uncertain you were about the overall benefit risk. And if their distributions were overlapping, you might not know which to choose. So what you do, essentially, is to go through and instead of forgetting your uncertainty once you start putting it together, you bring your uncertainty with you. So in terms of the measure, you could put a range on it. That would then propagate through, and so instead of a single number in that red box, which I'm afraid I can see it really clearly on the screen, I've realised you can hardly see it at all there, but there's a number in there, but you can see the distribution, I think probably rather more clearly. And then when you put those together using a sort of, you can call it a magic box, black box, or principled Bayesian statistics, depending on your language, what you get out for the example I've shown you is a distribution of overall benefit risk. And that blue one is what we think about placebo. That is the natalizumab, right at the right. And the other two coloured lines are the other drugs. And so the other drugs, to a large extent, we're not entirely sure that they are distinct from placebo. We think they probably are on balance, but they might not be. Whereas we are very certain that natalizumab is better. And that, as a statistician, gives me a lot more confidence in the statements I've been making than just the raw numbers. So we can do that. And if we do that, we can then do some other nice probabilistic statements. We can talk about the median overall balance. We can talk about what's the probability that outperforms placebo. We can talk about what's the probability natalizumab outperforms all other treatments. And that's actually kind of, pretty much 90%. So you can, if you do the formal mathematics, then you can present it in some really quite nice ways. So one of the dilemmas we had in this project is that certainly adds insight, but it is time-consuming, it needs expertise, and I have to say, regulators are saying, how are we going to do this? Companies are saying, what expertise do we need? And so what's appropriate depends very much on the problem. And this is one of the open research questions, actually, MCDA doesn't really account for the fact, at least not in a coherent way, that there are multiple outcomes, multiple studies, multiple treatments, and that is a very sparse matrix. And so Ed Waddingham, who we'd originally employed as a research assistant on the grant, and he'd done a lot of the work on it, is now doing a full-time PhD, which he's submitting either at the end of this year or beginning of next year, where what he's doing is some really nice Bayesian stuff and looking at some of the numerical stuff for dealing with that sparse matrix, allowing properly for the relationships, the correlations. Because the kind of question that was coming up was, well, some of the endpoints are very similar. You tend to have what's the proportion above this and what's the proportion above this cutoff. They're almost the same thing. And people say, aren't you double counting? I said, if you properly allow for the structure, then you wouldn't double count. Whereas if you don't properly allow for the structure, there's a danger that you might. So Ed Waddingham is doing that, and if you want to know more about it, you better ask Ed to come and give you a talk sometime. But what I want to talk about is that's fine, and it's keeping Ed very busy and he's thoroughly enjoying it. And it's sort of that bit of it is building on kind of existing methods. The bit that we did and realised we were only scratching the surface on was how we got those weight elicitations. Because they should come from patients or the regulators, and we'll come to the question about whose weights they are in a little while. But there's also the question of how you do it. And multi-criteria decision analysis has this sort of swing weighting, which is part and parcel of it. There's the beautifully named Macbeth, measuring attractiveness by a categorical-based evaluation technique. There's something called analytical hierarchy processes, and then borrowed straight from those very nice health economists, there's something called discrete choice experiments. That people say, well, that's what you want. So what we did during this project on this case study, and of course the other case studies, was to try some of these things out. And we didn't get as far as we liked, because of course you need ethics to do this, and so poster looked at about all the hurdles about doing research, we got into that as well. But we did do some, and some of them had to act surrogate because we had to do them with people available while we were trying to get our ethics. So what we're trying to do is to find the values associated with that little lot. So that's just what I've shown you before. And what I'm going to do is just give you a sort of, again, a top-level view of each of these techniques and broadly how they work, and if you want more, I'll point you to somewhere you can get more information later. So swing weighting, and typically with multi-criteria decision analysis, this is done with a group sitting around having this conversation, but it could be you sort of gazing at your own navel and coming up with it. And what you do is you take one of those groups of outcomes, so going back there, we've got a group up there, the group of serious side effects, and the group of less serious side effects. So if you take the serious side effects, then the first thing you do is rank them. So the group who did this particular thing thought, well, PML was the worst, and I think not many people would disagree on that. I think different people would quite legitimately permute the rest. But anyway, this group came up and said, well, the next one was reactivation of serious herpes virus, then seizures, and then the risk of abortion or congenital anomalies. And that harks back to the previous presentation about you've got two people, when you're dealing with pregnancy and you're trying to balance them. So if you happen to be pregnant, you might put that one higher. So you rank them. Then what you do is try and give them a relative importance. And that was what they came up with there. Arbitrarily, you give PML 100, and then you put the rest of where they are above 0. And that generates a kind of weighting. What you then do is you take the top one of each and use that for where to place these groups. And you do essentially the same thing between the top ranked one in each category. And then that, by scaling, just takes the rest with it. So it's a way of avoiding having to think through every possible comparison. So that's what swing weighting does. Macbeth is a much more qualitative technique. So what you're supposed to think is how much more attractive is it to move from worst to best for outcome I versus moving from worst to best for outcome J. And at each point you think, well, let's keep everything else equal. Then you sort of check the consistency of answers, because inevitably these things don't always come up with consistent answers. And then you compute your initial guess at the weights with some optimization, so numerical stuff there. And then you can refine them. And you come up with a ranking like that, and you put some qualitative judgments on it, and then the fine-tuning, the software compared with this gives you that, and you can have a play. And those lines say, this is the wiggle room you've got. If you go outside of those red lines, then you start being inconsistent with your original decision. The analytical hierarchy process, you go through sort of making pairwise comparisons. How much more important is this outcome than that outcome? And then you give it a number. And then, any mathematicians once upon a time here? Remember what your dominant eigenvector is? Anyway, you compute that and it somehow tells you the answer. And the value functions are computed in a similar manner. And essentially, swing weighting is quantitative, there are no consistency checks, weight calculation is very direct, you can see what you're doing with the weights. Macbeth is qualitative, you have to resolve your inconsistencies, and then it's got this linear optimization technique going on. And analytical hierarchy process is quantitative, you have to compute a consistency score, and you work with principal eigenvectors. So they've all got slightly different things going on. Discrete choice experiments work quite differently. You send out a questionnaire where you ask people to say, if we've got 2 treatments, number of relapse per patient, disability progression, 250 versus 100, PML 3 versus 0. If I just showed you that, I suspect most people would opt for A. But if I show you the second slide, we say, well, what if that one gives you mild allergic reactions half the time, and this one gives 200 out of 1,000 patients depression, whereas that one doesn't. And what you have to say for that question is, which treatment would I have? And then you get a swathe of these questions. Now, you can't possibly ask one person every combination. So this is done at a group level. The others can be done individually, but this is done as a group level. And basically, you send them out to loads of people, and each one gets a subset of the questions. And anybody who's done design of experiments in a previous life, of the kind of agricultural variety, will start to recognize things like split plot experiments. And you aggregate them. So, the reason it's difficult in health is that you've got so many outcomes with so many levels that numerically it just blows up. So you actually have to do quite a bit of reduction even to do the discrete choice experiment. So lots of questions about this, lots of methods, which do you use, whose preferences, is it the patient's, and this relates to another poster, who should we be looking at? How do you know that they're representative? How do you know that those weights I put up have any meaning at all? What do you do about multiple viewpoints? How much heterogeneity is there? And again, there are just enough of these things now coming out that you can think about beginning to do some meta-analysis and actually explore that heterogeneity. So that's the other part of his PhD. It's still very much work in progress. I think it does add value and legitimacy and so on, but lots and lots of questions about how you do it, feasibility, validity. And I'm delighted that IMI then commissioned another project to take this part of the work much further, because I was so conscious that we'd opened up a box, but we'd really not sort of bottomed it out. I could, but I won't because of time, say much more things about patient and public involvement. Some of this comes into doing things to patients, if you like, but a large strand of our work was actually how you work with patients and where they can input. And we did the review, we did a visualization review I've not talked about, we did several other case studies and a whole PPI work stream. And if you're interested in, and we then went through a recommendation roadmap, which is if you're coming in at particular stages, how can you best use this stuff? But what I want to say is, if you are interested with this, then We spent a lot of time on the website, working on a website, and all of this is in there. If you want to know about a technique or follow a case study through, that's what you should look at. And then finally, I just want to thank our funders, because without the EU funding, without the pharma funding, we couldn't have done any of that. So at that point, I shall stop, because I am having my traffic lights gone red.